Alright folks, welcome back to Blood. So this is the second blood lecture, and we are going to talk about red blood cells here. Alright, so first of all, we have a red blood cell count. It's an absolute count, and you can see the difference between men and women. They're about the same though. It's between 4 and say 6.5 million per microliter. Women run a little bit lower. The main reason women run a little bit lower is because they menstruate every month. That's one of the main reasons. Now that's an absolute count. That tells me how many cells per microliter of blood. But we also have another measurement called a hematocrit. And what this is a percentage of your whole blood. So what percentage of your entire blood is actually red blood cells? And I can tell you it's just under 50%. So again, women run a little bit lower. Women run from between 37 and 47%. 47%, of course, is just under 50%. And then men run just a little bit higher than women. They run between 40 and 54%. So men can actually go just a little bit over 50%. But the thing I want you to, the take home message, because in your life, you're going to be looking at a lot of RBC counts and WBC counts, white blood cell counts, and a lot of hematocrits. It's around 50%. And then think to yourself, okay, women are a little bit lower than men. So, and then think with women, I would say low 40s, and with men, I'd say high 40s. That's how I would think about it. Rather than memorize ranges, because ranges change from hospital to hospital, from lab to lab. Every lab sets up their own normal range. So that's how I would think about it. About 50-50, 50% cells and 50% plasma. And then say, okay, well, men and women are slightly different, so women are low 40s and men are high 40s. And that'll get you in the right range just thinking of it that way. Hematocrit is what we do is we actually draw some capillary blood right here into a microfuge tube, a microcentrifuge tube, sometimes called a microfuge tube. And we put a little clay in the end. This is a little clay, seals the end of it, and a little bit of clay here seals the end of it. And then we load these capillary tubes into a microfuge. This is a microfuge right here. Close the top, turn it on, it spins. When you take it out, you can simply measure the length of blood from here all the way up to here, and let's say it's 10 centimeters. Okay, well, the percentage of your whole blood that is red blood cells is how many centimeters the red blood cells go to. And let's say the red blood cells go to 5 centimeters. Well, 5 centimeters is 50% of 10 centimeters. And it really is as simple as that. So you do the same thing with the other patient here, a female patient in this case, and let's say it's 10 centimeters again. This is 10 centimeters all the way down through here. I'm using an arbitrary number, by the way. And then I measure the centimeters of red blood cells. And let's say it's 44.4 centimeters. Well, 4.4 centimeters is 44% of 10 centimeters. So the crit for this female patient would be 44%. So this is called a hematocrit, and it's a packed cell volume is what it is, measured in percentages. So I need you to know the difference between a red blood cell count, which is an absolute count of number of reds per microliter, versus a relative count, a percentage count. So what percentage of your whole blood is red blood cells? And it should be in the 40s, high 40s for men, low 40s for women. All right, so that's a red blood cell count. Please do memorize these red blood cell counts. Just think 4 to 6 million per microliter, and that'll encompass men and women. So you can see if you do 4 to 6, that covers the men, and if you do 4 to 6, that covers the women. So 4 to 6 million per microliter. And by the way, remember in your book might say cubic millimeters. A cubic millimeter equals a microliter. They are equivalent to each other. All right. This is a picture of a red blood cell. Let me give you some characteristics that you should memorize. First of all, a red blood cell is about 90 femtoliters big volume. By the way, you're going to say, what's a femto? Well, you have 1 liter, and 1/1000th of that is 1 milliliter. And 1/1000th of a milliliter is a microliter. So a microliter is 1 millionth of a liter. 1/1000th of a microliter is a nanoliter. 1/1000th of a nanoliter is a picoliter. And 1/1000th of a picoliter is a femtoliter. femtoliter. So if this is 10 to the negative 3, and that's 10 to the negative 6, and that's 10 to the negative 9, and this one's 10 to the negative 12, a femtoliter is 10 to the negative 15. So a femtoliter is 1 over 10 to the 15 of a liter, of 1 liter. A femtoliter equals 1 over 10 to the 15 of 1 liter, or 10 to the negative 15 of a liter. 10 to the negative 12th is a trillion, 1 trillionth. 10 to the negative 12th is 1 trillionth. Therefore, 10 to the negative 15th is 1 quadrillionth. That's how small red blood cells are. And of course, you can't see them with the naked eye. You have to look under a microscope. So they're about 90 femtoliters volume. They live about 90 to 120 days. So you see, you got a 90 here and you got a 90 here. You can think, okay, they're 90 femtoliters. They live 90 days. So you can think of that. They're about 7 micrometers diameter. So if I measured from here to here, that would be about 7 micrometers. Just small enough to fit through capillaries. Matter of fact, capillaries are sometimes 5 and 6 micrometers. Well, how could that be? How could a 7 micrometer cell fit in a 6 micrometer capillary? Because they can squeeze. These guys can squeeze down. Matter of fact, if a red blood cell is not deformable, if it can't change shape and squeeze through the capillaries, then we have a problem. And there's some diseases where they can't. Red blood cells don't have a nucleus. They extrude it before they leave the bone marrow. I mean, they're in their lineage, in their development lineage, they had nuclei, but the mature red blood cells lose their nucleus before they leave the bone marrow. All right, so that's a red blood cell. Oh, and it's a biconcave disc. Concave means it makes a cave, and it's a biconcave because there's a cave on the bottom and there's a cave on top. So it's a biconcave disc. So this is a red blood cell. Primary function of all red blood cells, carry oxygen. All right. These discs bend and flex. They bend and flex. They have to be able to fit through the capillaries, which are slightly smaller than the red blood cell. You look at right here. This is your book's telling you that some capillaries are as small as 4 micrometers. Well, that makes the red blood cell almost twice as big as it. So it has to be able to bend and flex. All right. Now this word rouleau. Let me go back. Rouleau just means stacking, like a roll of quarters. When these red blood cells squeeze through the capillaries, they make these rouleaux. You can see how the red blood cells are stacked almost like a roll of quarters. That's called rouleau. Now, rouleau is normal if you're squeezing through the capillaries, but if you see rouleau in the big arteries and big veins, that's abnormal, and that's indicative of inflammatory responses. So lab techs actually look for rouleau when they look for abnormal rouleau, I should say. All right, so that's red blood cells in a nutshell. This is why they can carry oxygen. Red blood cells have this protein called hemoglobin. All right. Now, the hemoglobin is the holoprotein, but it's not made of just all protein. The apoprotein is called globin, and the prosthetic group is called heme. And when you put the prosthetic group with the protein, you get hemoglobin. Well, this prosthetic group right here, this heme, is not proteinaceous. It's organic, but it's not a protein. So this is heme. And at the center of the heme molecule is an iron. And by the way, the fact that that iron is positive 2 is very important. It's called ferrous iron. And iron plus 3, called ferric iron, doesn't carry oxygen. So our bodies have many mechanisms to keep our iron in our heme at the plus 2 oxidation state, the ferrous state. Can that ever fail? You bet. There's a bunch of diseases where we can't keep our iron in the ferrous state, and it's in the ferric state. Methemoglobinemia is one of them. We'll talk about it a little bit later. So it's important that hemoglobin has proper heme. It's important that the iron of the heme is in the ferrous state, and where oxygen actually binds is oxygen binds to the iron. So I'm just going to stick the oxygen on the iron right there. I drew a long line. It's not really like that, but I had to have space to draw O2. So oxygen binds to the iron of hemoglobin, which is in your red blood cells. The globin part of hemoglobin has 4 polypeptide chains. It has 2 alpha chains. Here's alpha chain 1 and here's alpha chain 2. They're denoted dark purple in this picture. And there's 2 beta chains, beta chain 1 and beta chain 2. They're denoted light blue. So sometimes we say hemoglobin's alpha 2, beta 2 because there's 2 alpha chains and 2 beta chains. Each polypeptide chain carries their own heme. Therefore, there's 4 hemes per hemoglobin because the alpha chain 1 has a heme, alpha chain 2 has a heme, beta chain 1 has a heme, beta chain 2 has a heme, and therefore each hemoglobin molecule can carry 4 oxygens. So there you go. You want to know how many oxygens can a hemoglobin carry? One hemoglobin molecule can carry 4 oxygens. And there's lots and lots of hemoglobins per red blood cell, and there's lots and lots of red blood cells in your body. How many? 6 million per microliter. So how many total? I don't know. Take 6 million per microliter and multiply it by how many times a microliter goes into, say, 5 liters. So a microliter goes into 1 liter 10 to the 6 times. So it's 5 times 10 to the 6. So if you take 6 times 10 to the 6 times 5 times 10 to the 6, you will get 30 times 10 to the 12th. There's 30 times 10 to the 12th hemoglobin molecules in your body. That's 30 trillion of them. That's how many hemoglobins in your body. Now, that's just academic. I'm not going to ask you to calculate that. I'm just showing you that, yeah, we can figure out how many hemoglobins we have in our body. And in fact, we measure hemoglobin in our blood. We measure hemoglobin in our cells. We actually calculate it. It's a calculation. And there's names for these measurements in the lab, and you'll learn some of these in your life. I actually might talk about a couple of them here today. All right, so that's hemoglobin carrying your oxygen. Notice that with males, we run a slightly higher hemoglobin than females. Males run around 14 to 18 grams per deciliter. That's how much hemoglobin a male has. I usually remember about 16. And then women run between 12 and 16 grams per deciliter. I usually remember about 14. So I hardly ever remember ranges. I always remember that there are ranges, but I have a hard time with ranges, so I just remember one number that's in the middle of the range. So men are about 16 grams per deciliter hemoglobin, women are about 14 grams per deciliter hemoglobin. If you don't have enough hemoglobin, you are anemic. By the way, if you don't have enough iron, you are anemic. By the way, if you don't have enough red blood cells, you are anemic. So you can see that there's different types of anemia. There could be anemia of low blood cells, red blood cells. There could be anemia of plenty of red blood cells, but not enough hemoglobin. There could be anemia of plenty of red blood cells, plenty of hemoglobin, not enough iron. So you can see that there's different diseases going on here with our oxygen carrying capacity. All right, that's hemoglobin for you. When hemoglobin binds oxygen, we call it oxyhemoglobin. Matter of fact, this is showing you deoxyhemoglobin right here. And when it binds oxygen, we call it oxyhemoglobin. In the arteries, about 99% of our hemoglobin is carrying oxygen in our arteries. In our veins, 40 to 50%. So veins aren't completely devoid of oxygen. About half of the hemoglobin in our veins carries oxygen, but just about all of it in our arteries does, 99%. That's called an oxygen saturation, by the way. When you measure how much oxygen the hemoglobin's carrying, it's called an oxygen saturation. Arteries are 99% saturated, and veins are only about 40 to 50% saturated. All right, a couple other things. CO2 can actually bind hemoglobin, but CO2 binds the protein part of hemoglobin, the globin part of hemoglobin. It doesn't bind the heme. So CO2 is not competing with oxygen. We call this hemoglobin carbaminohemoglobin if CO2 is bound to it. And about a quarter of our CO2 travels bound to hemoglobin. Most of our CO2 floats around our body as bicarb. And here's a very important thing you have to know. If CO2 combines with water, you get carbonic acid. And there's an enzyme that does that called carbonic anhydrase. And then anhydrase. Carbonic anhydrase. Let me spell that correctly for you. It's actually spelled correctly. It's just horrible handwriting. Horrible, horrible handwriting. My mother would be ashamed. Hydrase. Carbonic anhydrase converts water and CO2 to carbonic acid. This is carbonic acid right here. And that spontaneously deprotonates like all acids do. This is the definition of an acid, that it deprotonates like this. So really what we're doing is this CO2 is being carried around as this bicarb. 70% of it does. 70% of it. These reactions are reversible, by the way. All right, so we have oxyhemoglobin, we have deoxyhemoglobin, we have carbaminohemoglobin. We know how most of our CO2 is being carried. 70% of it's carried as bicarb. All right, so look at this. There is some oxygen dissolved in our plasma, but only about 1%. Oxygen doesn't dissolve well in aqueous solutions. CO2 dissolves a little bit better. About 7% of our CO2 is dissolved in plasma. Now, why do I mention that even? Well, first of all, when I get to the respiratory chapter and I tell you why you breathe, like what's driving you to breathe, the answer is going to be these gases that are dissolved in the plasma. If you have a lot of oxygen dissolved in your plasma, well, when I say a lot, it's always going to be around 1 to 1.5%, but it's in equilibrium with the oxyhemoglobin. If you saturate all the hemoglobin, then you'll have a full 1.5% dissolved O2 in your plasma. If you start unsaturating your hemoglobin, then that 1.5% will drop. So what can cause you to breathe is oxygen and CO2 concentrations. And by the way, CO2 is a more powerful respiratory drive than O2. But we'll talk about that in chapter 23. So if you're, let me just make it simple for now until chapter 23. If your CO2 is high, your brain tells your diaphragm to contract. You breathe. If your CO2 is low, your brain doesn't tell you to breathe as quickly. You'll have a resting breathing rate if your CO2 is low. If your CO2 is high, you'll have an active breathing rate. And oxygen plays a role as well. This is going to show you how hemoglobin helps us dissolve more, well, not dissolve it, but carry more oxygen. What I'm going to do is add oxygen to the solution, and then you'll see the concentration of oxygen in solution goes down as it binds hemoglobin. So, well, there's hemoglobin right there. And now look at the oxygens bind to it. Now there's fewer oxygens in solution. See, now there's only 2 oxygens in solution. This one right here and this one right here. The only 2 oxygens in solution. So now who has a higher oxygen concentration, A or B? And the answer is A does. So actually the oxygen is going to go towards B now. And as long as I can keep binding hemoglobin, I can carry more oxygen. Because this solution over here could be saturated with oxygen, but when it's carried on the hemoglobin, the solution doesn't have it. The hemoglobin has it. So by having hemoglobin, it allows us to carry more oxygen. And that's pretty cool that our blood does that, because our plasma only can carry 1.5% oxygen. That's all that can be dissolved in the plasma. All right, so that's hemoglobin. That's hemoglobin binding oxygen. Now, red blood cells only live 90 to 120 days. Look at this, average lifespan of red blood cell, 120 days. I told you 90 to 120 days. Your book will say something like that, because I wanted you to have something easy to remember, like red blood cells are 90 femtoliters, and they live 90 days. I usually remember they live 120 days, but you can use 90. It's a range. When the red blood cell dies, we recycle the hemoglobin. Now this is pretty cool. The heme we turn into bilirubin. A macrophage converts the heme into bilirubin. That macrophage delivers the bilirubin to the liver, and that becomes part of our bile and helps us digest fat. All right. What do we do with the globin? The globin is converted to amino acids, and these are recycled. We use them. What do we do with the iron of the heme? This is carried by transferrin to the liver and bone marrow and stored in ferritin. Ferritin is a storage protein for iron. So transferrin carries the iron to the bone marrow, or liver it could be, by the way. And then these protein molecules inside our bone marrow stores the iron. And then it is used for new heme synthesis. So we are pretty conservative with our iron. But it doesn't mean you don't need a little bit of iron each day in your diet. You do. Because you're going to lose some iron in your feces. You're going to lose some iron in your urine. But we do conserve most of it. All right, so this is the breakdown of hemoglobin into heme and globin and iron and what we do with each. Look what happens to the bilirubin right here. First of all, it's put in bile, and then it's put into our intestine, and then bacteria in our intestine convert the bilirubin to urobilinogen first, urobilinogen, and then from urobilinogen to urobilin, which is yellow, and that goes to my kidneys, and that's why my urine is yellow, because of urobilin, and stercobilin, and that's brown, and that's why my feces are brown, because of stercobilin. So why my urine is yellow and why my feces is brown has everything to do with bilirubin and bilirubin breakdown. So there you go. Bilirubin becomes urobilinogen, urobilinogen becomes urobilin and stercobilin. Urobilin is yellow, I urinate it out. Stercobilin is brown, I excrete it in the feces. Now, you're going to learn a bunch of diseases in your life about blocking the common bile duct, about liver cirrhosis, and about things like this. And the bottom line, when your liver doesn't work or you block your bile duct, is this bilirubin builds up in your bloodstream. Well, why wouldn't it? You are blocking the bile duct. You can't put it into the intestine. You are cirrhotic. You can't process it. You can't conjugate it and put it into your intestine. So bilirubin builds up in the bloodstream. That makes you jaundice because bilirubin has a yellow color to it. And jaundice is when you have high bilirubin in your bloodstream. Okay. This is a same picture. Same picture. By the way, with these two words right here, hemoglobinuria, if your red blood cell destruction is too fast, exceeds its normal rate of that your body can handle, that means these macrophages here can't handle all of the hemoglobin that's being delivered to it because of increased red blood cell destruction. What you get is hemoglobin in your urine, and that's called hemoglobinuria. You could actually get red blood cells in your urine, and that's called hematuria. Hemoglobinuria and hematuria are similar but different. Hemoglobinuria is hemoglobin in your urine, and hematuria is red blood cells in your urine. So that's the difference between those two.
When old red blood cells rupture, the released hemoglobin is ingested by macrophages. The globin chains of hemoglobin are broken down to individual amino acids that are metabolized or used to build new proteins. Iron is released from the heme of hemoglobin. The remaining structure is converted to biliverdin, which is then converted into bilirubin. Iron is transported by transferrin in the blood to various tissues for storage, or to red bone marrow for making new hemoglobin. Free bilirubin is transported by albumin in the blood to the liver. Liver cells make conjugated bilirubin, which is excreted as part of the bile into the small intestine. Intestinal bacteria convert bilirubin into bilirubin derivatives, which contribute to the color of feces. Some of the bilirubin derivatives are absorbed into the blood and excreted from the kidneys in the urine.
All right, so that's the bilirubin cycle. Now, let's look at the red bone marrow. This is making a red blood cell, and you can see that it starts out with a blast cell, and it's called a proerythroblast. So this is your bone marrow. You got a proerythroblast. This is going to differentiate into a mature red blood cell. So here's the differentiation pathway right down through here. By the way, you could have differentiation without maturation that leads to a disease. And you could have, can you have maturation without differentiation? Oh, let me think. Maybe so. I'm thinking that maybe it could happen, but I haven't heard of that happening too much. So what happens is the proerythroblast differentiates into an erythroblast. All right, and that's the second day of this production. By the way, look at this. It takes about a week to make a red blood cell. The erythroblast then differentiates further, matures now. By the way, I've already differentiated now. This cell is destined to be a red blood cell. It cannot become anything else, so I've differentiated. Now all it has to do is mature into a mature red blood cell. So the erythroblast becomes the polychromatophilic erythroblast right here. The polychromatophilic erythroblast becomes a normal blast. We call this normal blast a nucleated red blood cell. That's what we call that. Nucleated RBC. Sometimes the bone marrow releases these nucleated RBCs into the peripheral circulation. And I've seen them before doing a differential, doing a smear differential in the lab. You don't see a lot of them. You see some of them. You see them occasionally. You can see a lot of them with abnormal conditions, some cancerous conditions and things like that, or an overactive bone marrow. Usually, however, the normal blast extrudes the nucleus, and it's a reticulocyte briefly. And what that means is it has this basophilic staining pattern inside of it. That goes away. That's just some leftover RNA. That's all that is. That goes away, and then the reticulocyte relieves the bone marrow. You can actually see some reticulocytes in the peripheral blood. So that relieves the bone marrow and finishes its maturation. You don't see many reticulocytes in the peripheral blood. So right about the time that the reticulocyte's leaving the bone marrow is about the time it's finishing up its maturation. But you can sometimes see some retics in the peripheral blood. Now, what turns this whole thing on? And the answer is a hormone called EPO. It's called erythropoietin. This is a hormone from your kidneys that floats to your bone marrow and says turn on red blood cell production. And red blood cell production itself is called erythropoiesis, promoted by the hormone called erythropoietin. So erythropoietin causes erythropoiesis. And that's pretty cool. The words are very similar, so you can... And erythropoietin is the EPO that you hear about on TV when some Olympic athletes abuse it. Because, you know, you could go to Denver, Colorado and train in low oxygen environment, and your body would naturally release EPO. So you would naturally produce more red blood cells, and therefore you'd be able to run the marathon in a shorter period of time. However, some people don't want to move to Denver, Colorado. So they live at sea level, and they take a shot of EPO every day, or every once a week, or I don't know how you abuse EPO. I don't know how many times you take a shot, but you take EPO and you artificially raise your red blood cells without the low oxygen tension. So there's red blood cell production.
Hemopoiesis is the process of blood cell formation, which occurs primarily in the red bone marrow. Hemopoiesis starts with undifferentiated cells called hemocytoblasts. These stem cells give rise to the many different cell types that make up the cellular elements of blood. A number of hormones and growth factors, known as colony-stimulating factors, or CSFs, influence hemocytoblasts to differentiate along either the myeloid or the lymphoid cell line. The myeloid cell line forms erythrocytes, or red blood cells, and megakaryocytes, which give rise to platelets. Myeloid stem cells also form leukocytes, or white blood cells, except for lymphocytes, which are produced by the lymphoid cell line. Hemopoiesis along the myeloid cell line follows one of three pathways: erythropoiesis, thrombopoiesis, or leukopoiesis. Erythropoiesis produces red blood cells. It begins with stimulation of myeloid stem cells by CSFs. This produces a progenitor cell that gives rise to a proerythroblast, the first committed cell along this pathway. The hormone erythropoietin initiates the erythroblast stage, during which proerythroblasts are stimulated to form erythroblasts. The erythroblast begins to synthesize hemoglobin and gradually decreases in size, giving rise to a normoblast. At this point, the nucleus is ejected from the normoblast, resulting in a reticulocyte. After the reticulocyte enters the bloodstream, its organelles degenerate, at which point it is considered a mature erythrocyte. Thrombopoiesis produces platelets. Like erythropoiesis, it also begins with a myeloid stem cell that produces a progenitor cell when stimulated by growth factors. The progenitor cell becomes a committed cell called a megakaryoblast. In response to the hormone thrombopoietin, the megakaryoblast differentiates into a large cell called a megakaryocyte. Platelets are formed when thousands of small cell membrane-bound portions of the cytoplasm break away from the parent cell. These isolated fragments enter the circulation as platelets. Leukopoiesis, the formation of white blood cells, involves maturation of 3 different cell types. The myeloid cell line gives rise to granulocytes, eosinophils, basophils, and neutrophils, and monocytes. The lymphoid cell line produces lymphocytes. Granulocytes and monocytes share a common stem cell. In granulocyte maturation, the stem cell is stimulated by CSFs to produce a myeloblast, which then gives rise to promyelocytes. These cells form the 3 myelocyte subtypes. Eosinophilic myelocytes differentiate into eosinophils. Basophilic myelocytes differentiate into basophils. Finally, neutrophilic myelocytes develop into neutrophils. The common granulocyte stem cell also participates in the formation of monocytes. This stem cell will give rise to a monoblast that differentiates further into a promonocyte and then a monocyte. The lymphoid stem cell is also involved in leukopoiesis, which results in lymphocyte formation. This stem cell differentiates into B lymphoblasts and T lymphoblasts. These mature into B and T lymphocytes, respectively.
All right, there's one concept I want to add to that. Let me actually just show you on the video. It's the concept of differentiation and maturation. That's the concept. So let me show you right now. This myeloid stem cell right here, this myeloid stem cell has to differentiate, because right now it's pluripotential. That's pluripotential. It means it has the potential of becoming more than one thing. Because as you can see, it could become a platelet, it could become any of the 4 of the 5 white blood cells. It could become lots of things. So it's pluripotential. If it differentiates into an erythropoietic stem cell, it's differentiated. This cell here doesn't become anything other than a red blood cell. So from here down through is just maturation. Whereas from the myeloid stem cell to the erythropoietic stem cell, it's differentiation. So there's a difference between those two words, differentiation and maturation. So that's the difference between differentiation and maturation. This is a table showing you some the crit, the retic count, and things like this, and it's showing you some abnormal values that are associated with different diseases. So if you had an elevated crit, what could you have? You could have polycythemia, like polycythemia vera. You could have erythrocytosis caused by lots of things. One of them is cancer, by the way. You could have leukocytosis. What if your crit's low? You could have any of several types of anemias, and so on and so forth. I'm not going to go down through and read all these to you. So this is a table showing you normal values and elevated versus decreased values. You're going to ask me, how do I ask that on a test? Well, typically what I do on a test is I give you the normal values, and then I make you analyze what it means to be abnormal. Now, that doesn't mean memorizing 210 disease states. It means analyzing what it means physiologically. So without memorizing a bunch of diseases, you take a known normal, and you take an abnormal value, and you answer a question. All right, so that's just that. That's the next part is clotting. No, actually, I take that back. The next part is blood typing. red blood cell blood typing. Extremely, extremely important. So I'll see you in the next lecture where I'm going to beat into your head blood typing. Here's why it's so important. When you go to the lab to get units of blood to hang on your patient, that's the one thing you can do that can kill a patient very, very quickly. So the lab has to know what they're doing, and the nurse has to know what they're doing, so you hang the right unit of blood on the right patient. All right, so next lecture I'll be covering slowly and in detail.